I'd like to thank the organisers, particularly Dr. Guy Betts, for the opportunity to be able to speak in the symposium today, and also to speak with other esteemed experts in the field. It's good to speak with Eddie and Sunada today. The biology of potentially malignant oral lesions, PMOL, is very close to my heart. It's where I started my research about 20 years ago, and in that time there have been a mountain of literature, but only relatively modest success in being able to translate this into clinically useful changes. The aims of the experimentation that we're going to talk about, of course, are well understood. We're looking at understanding the disease. We're wanting to help in our prediction of malignant transformation, and also hopefully to be able to identify new targets for therapy. And it's very much with patients in mind that we try to make progress in this field. There has been much interest in the molecular pathogenesis of head and neck cancer or oral cancer. A lot of the data which is available is focused upon malignant disease, but there is increasing data on the early stages of the development of these cancers, including potentially malignant oral lesions. Many years of experimentation have been summarized in a number of graphics, as you can see in the one on the screen, which has come from Benny Lehman's 2011 paper in Nature Reviews in Cancer, and includes very many of the well understood cancer pathways. HPV is included here, but I'm not going to spend any more time talking about HPV. It's a very interesting subject in its own right. But we're going to see later, one of the main problems in this is that there is significant molecular heterogeneity, which characterizes oral squamous cell carcinoma. And it's a big issue in translating these molecular schema into something which is practically useful for our patients. So with this in mind, we have a vast amount of ground to cover in this talk. And thus I'm going to focus down on to the headlines and the areas pertain to PMOLs, looking at different techniques that have been used to assess them, moving all the way from the whole cell, the whole DNA or content of the cell, all the way through to detailed transcriptomics and proteomics in these lesions. One of the main underpinning and facilitating abnormalities in the development of many cancers is genomic instability. And as many lesions progress, there are a number of mechanisms by which this could occur. On the left here, we see one schematic which relates to the escape of cells from replicative senescence, and they will then pass through crisis and become replicatively immortal. But that process results in considerable genomic instability. Many cells die, but a small proportion do survive, and they have quite substantial genomic abnormalities. But there are other ways in which genomic instability can arise, including abnormalities in chromosomal segregation and in the construction of mitotic spindles. Now, these processes result in very complex alterations to the genome at the level of the karyotype, chromosomal loci, and also individual genes. And in many cases, it can be quite challenging to identify which of these have resulted from the genomic instability and are thus relatively non-specific, and which of these are true key alterations. And an illustration of this, I think, is the fact that we have found the identification of driver mutations in head and neck squamous cell carcinoma to be quite difficult. At the level of the DNA content of the cells, assessment of ploidy has been undertaken by a number of groups, including those in the UK and in Canada. The underlying principle is that the overall DNA content of the cells will become altered, and this can be detected by image cytometric techniques. And so the alterations in the DNA content of the cells, which are non-modal, it's not an exact multiple of the normal modal number of chromosomes, are significantly abnormal. These are referred to as aneuploid cells. And you can see in the image here, there's an array of segregated nuclei which are stained. They are then scanned and assessed. And the level of ploidy, the level of abnormality within the DNA content of the cell can be ascertained. What about the clinical use of these? Well, it's provided some predictive data on malignant transformation, and I point you in the way of Eddie Odell's very useful review of this year in oral diseases for a wider dealing with this topic. And some analysis demonstrate very little malignant transformation in diploid lesions, significant malignant transformation in aneuploid lesions, whereas others, for example, in the center here, just taking the ploidy status shows that there's actually quite a significant malignant transformation within the diploid or tetraploid population. This has resulted in those working, for example, in the paper by Saliva et al. From last year, looking at combining this with dysplasia grading. And that has been shown to increase the diagnostic yields on this. And so when you have an overall combination of ploidy and dysplasia grade into high and low risk, you get quite a marked separation between malignant transformation in those which are in the high and low risk groups. Moving on to a more focused group, down into the genome itself and copy number alterations. The first of these is loss of heterozygosity. Investigation of patterns of loss of heterozygosity at specific chromosomal loci have revealed rather consistent patterns of LOH. These changes vary between the loci. Some might be copy number neutral, some are associated with copy number loss. The loci of interest have been well rehearsed. There was at chromosome 9p, CDKN2A locus, 17p, which is the p53 locus. And there are others, as you can see in the diagram here, which are indicated whose specific gene locus association is becoming understood. Some are still a little elusive. And these changes, they accumulate during the progression of potentially malignant oral lesions into oral squamous cell carcinoma. And in fact, the loss of heterozygosity patterns have been the basis for many of the molecular models of oral squamous cell carcinoma, all the way back to the initial large-scale one by Joe Califano of many years ago. But the knowledge of loss of heterozygosity has been incorporated into risk models. And for example, from the British Columbia group here, they're demonstrating the use of loss of heterozygosity in cohorts of oral epithelial dysplasia patients. Progression of potentially malignant oral lesions occurred in 53.6% of those who had a high-risk LOH profile, that is alteration at 9p, 17p, and 4q. Whereas those who didn't even get off first base, who'd retained 9p, the malignant transformation rate was 1.5%. And these molecular risk-based models are still largely used within research context. So the British Columbia group, I think, are probably the furthest ahead in implementation of such analyses into more routine clinical usage. Looking at the overall pattern of alterations in the genome, now not just focusing on specific loci, which we can do using techniques such as array CGH or low read depth next generation sequencing, these demonstrate a much wider range of alteration within the PMOL genome. This figure here shows some of our work that's been done in the panel of British Beatson Institute of Cancer Research cell lines, and the patterns of copy number alteration in the oral squamous cell carcinoma cells on the right-hand side. You can see that there's changes either gain or loss on most chromosomes. But if you have a look at the left-hand side, you can see that it's also reflected within the PMOL cells as well. Fewer number of alterations, but the pattern is largely the same. So what happens when you start to have a look at that in clinical cohorts, which are that bit more complex? And the paper by Bhattacharya et al. From a decade or so ago looked at that, and they could demonstrate that, in fact, a very similar thing was demonstrated. In terms of the clinical cohorts, you can see that there are changes, many changes in the squamous cell carcinomas, but the site in the dysplasia cases was very similar. Fewer numbers, as we saw in the cell lines, but a very similar pattern of chromosomal loci, which are identified as being altered. Interestingly enough, when you then look at the PMOLs and split them into those with high-grade and low-grade dysplasia on the binary scale, and this is a group which was published by the Leeds group a number of years ago, the differences shown between low-grade epithelial dysplasia and high-grade epithelial dysplasia are quite evidently seen. There are fewer changes in the low-grade dysplasia. But even still, that pattern of changes can still be recognized within the low-grade dysplasia group. 3p, 3q, alterations in chromosome 8. And in terms of high-grade dysplasia, the vast majority of changes which are seen in high-grade dysplasias are also those which are seen in the invasive lesions. Overall, there's been substantial progress made in the assessment of the mutational landscape of head and neck squamous cell carcinoma over the past decade or so. The publication of the 2 papers in Science in 2011 started this and gave detailed insight into this and demonstrated further the molecular heterogeneity of head and neck squamous cell carcinoma. And in the updated 2015 Cancer Genome Atlas Network paper based on TCGA data, you can see here from the panel on the left-hand side of the slide that only p53 was mutated at a high rate within these samples. In fact, there were really no other single gene above about 20–25% of the samples. Alterations of those, for example, in CDKN2A, we've already talked about that, and we know that these are very well established. But others such as FAT1 and PIK3CA, and interestingly Notch1, which I'm going to come on to talk about, are of interest. Of course, this data refers to mutations in single genes, and there may be similar effects to be gained by alterations at other points in pathways, and there needs to be more sophisticated analysis to have a look at how the overall pathways are affected by the pattern of mutations which we see. In potentially malignant oral lesions, the data is more scarce. In vitro, in the cell lines again, the beats and cell lines, we found similar alterations to those which were seen in the squamous cell carcinomas. And a couple of analyses of tissue samples, again from the Leeds group, showed that at a very early stage of the development of these potentially malignant lesions, P53 was the only gene that was frequently mutated. And similarly, in the abstract from ASCO for a couple of years ago by William et al., P53 most common, and 3 other genes which I've already mentioned being present has been mutated in these samples. P53 alterations are very common in a number of cancers and also in head and neck squamous cell carcinoma. And P53, as has been well rehearsed, affects a number of very important cellular processes in terms of the functioning of the cell cycle, proliferation, migration, cell survival, entry into apoptosis, and so on. And there's been a lot of interest over many years of alterations in P53, and the mutations have been identified in PMOLs. A lot of the initial investigations centered on immunohistochemical analysis of P53 expression. P53, when mutated, in some mutations largely, it becomes stabilized. And so you get an increase in the proportion of cells which are expressing P53 and an increase in intensity. But despite the voluminous literature, in fact, this is an insufficiently sophisticated analysis to give us to link P53 mutation to clinical outcomes and malignant transformation. There are other abnormalities which may also be important, and these cannot be picked up by immunohistochemistry. Interestingly enough, the group at VUMC in Amsterdam many years ago noted that you get very small patches of P53 alteration which can be seen in immunohistochemistry widely in areas of field change, and some of these mutations and these abnormalities could be seen in later PMOLs or in fact even in squamous cell carcinomas themselves. Notch1 is of more recent interest, and as I say, the more recently identified mutations has been attracting attention. And perhaps it's more common than the next generation sequencing data has indicated. And for example, the paper, which you can see here in Cancer Prevention Research from a couple of years ago, shows that there may be abnormalities in up to 50 or 60% of PMOLs and squamous cell carcinomas. The mutations in Notch1 tend to be loss of function, and they alter cell fate and remodeling of the stroma, things which will promote tumorigenesis and affecting the intracellular signaling pathways of Notch1. Now, given the widespread genomic alterations that we see in PMOL, both in copy number alterations and mutations, it's not surprising that there are profound changes in the transcriptome, both of PMOLs and of head and neck squamous cell carcinomas have been reported. And these have been extensively analyzed both in vitro and also in vivo. What has been demonstrated is that PMOLs share many of the transcriptomic changes which are present within the squamous cell carcinomas. And on the left-hand side of this slide here, you can see this is our in vitro data, again from the cell lines, and particularly looking at the right-hand half of that figure, where you can see a large blue oval and a large red oval. And these are our immortal, potentially malignant cell lines and our immortal squamous cell carcinomas. And you can see the overlap, which you can in the overall transcriptome pattern of gene expression. And similarly, then when you take that into a whole tissue context in the paper by Makarev from a few years ago, you can see again that there's significant overlap. And if you have a look at the band at the top of the green and the red, the green being the leukoplakias, you can see that there are quite a lot of them mixed in with squamous cell carcinomas. It demonstrates that there is quite a significant overlap in the transcriptome, which is common between the PMOLs and the oral squamous cell carcinomas. However, validation of all these transcriptomic analysis and then translation of it into useful biomarkers has been very slow. And it has also been hindered by the molecular heterogeneity of a lot of these lesions. In oral squamous cell carcinoma, work has demonstrated a few years ago some molecular subtypes from this data. There are 6 of them, and that does dictate or is associated with clinical outcome, with the HPV-like one being associated with best outcome, and classical smoking or immunoreactive subtypes being associated with poorest clinical outcome. A recent paper taking these and looking at them in PMOL data, in the Carenzo et al. Paper from last year, again shows that there's an impact of these particular subtypes on the malignant transformation, the progression of PMOLs. And separating out these individual subtypes may allow us to identify useful biomarkers which will define or identify a particular risk. There are also other predictive structures, signatures, subtype and pathway signatures in PMOL. Some of these have come from the data which I have just shown, and others, for example, the 12 gene pathway from the William et al. Abstract from a couple of years ago. And you can see that taking a panel of about 12 or 13 genes does give us quite a separation between those who have got high and low risk of progression of the malignant, of their PMOLs. What about proteome? Moving on from nucleic acids into proteome. Well, tandem mass spectrometry has been used in a lot of different circumstances, be it in cell culture, in tissue samples, and also, interestingly, in saliva. And there have been a number of analyses in various contexts in relation to the progression of PMOLs to squamous cell carcinoma. A paper by Rauhan from a number of years ago now showed a lot of abnormalities within particularly important cellular networks. But this again has been slow to find its way into useful biomarkers which will be helpful clinically. There's been a lot of interest in progressing some of these proteomic markers into saliva diagnostics, and again using mass spec to look at variations in some interleukins, IL-6, IL-8, matrix metalloproteinases, and also looking at metabolites of these cells, which may be useful information. But also saliva is a much more useful analyte in terms of looking at DNA and RNA, microRNAs. A number of them have been identified as being altered in saliva and long non-coding RNAs. And there's particular interest in the role that extracellular vesicles may play. Now, these can transfer nucleic acids and proteins between cells, and these are being investigated, as are these other analytes in relation to saliva diagnostics, with the overall hope that we might get a saliva-based test which would revolutionize early diagnosis of PMOLs and squamous cell carcinoma. So what about moving forward? There are a lot of outstanding questions. There's a huge volume of scientific literature, and I've scratched the surface of it, but there's so few biomarkers that actually made it through into the clinic. So how can we use all this data? We still need to identify the initiating events and the drivers. That may help, even if they do exist. And of course, it may be that they are different within the different subtypes, as I've shown. But also laying it alongside this, and a completely different subject to an extent, is the integration of image analysis into this with the biology and using artificial intelligence. And maybe the most powerful way forward with this is going to be a combination of the biology and automated analysis of the morphology, we shall see. But the end of it, not only in diagnostics, is that we might hopefully get some therapeutics. There is a real need for medical treatment of PMOLs, as they're extensive, and in many cases, they're not open to surgical removal. So laying aside the whole area of retinoids, which we could spend a whole session talking about, there has been some work ongoing related to the molecular features of these lesions. And the EPOCH trial looked at using erlotinib, which is an EGFR inhibitor, as a chemoprevention in patients who have high-risk PMOLs with the patterns of LOH that we've talked about earlier. And the finding of this was that erlotinib really does not reduce malignant transformation. And in fact, the EGFR expression and status does not predict response, and that was rather disappointing. However, there is more on the horizon, and this rather serendipitous observation that valproic acid, which has been given to military veterans in the US for years for treatment of a number of different conditions, reduced head and neck squamous cell carcinoma development was really quite striking. And a phase 2 trial of valproic acid has opened in the UK in PMOLs to have a look at this effect. And this is being led by Richard Shaw in Liverpool. Now, in this context, the mechanism of action of valproic acid is not clear, but it may be via histone deacetylase inhibition. We've not even mentioned the whole area of epigenetic alterations thus far. And so less specific treatments, which can alter the expression of a number of different genes, may also be proven to be successful and useful strategies in treating these lesions. So there are a number of people who I'd like to thank. I'd like to thank, first of all, again, the organizer for the opportunity to present in this symposium. And there are many, many people to thank who have worked on over many years in this area. And those mentioned on the slide are those who have a direct impact into the work which I've shown and the views that I've expressed today. And I'm obviously very happy